My name is Marco Sardiello, and I work as assistant professor at the Department of Genetics at Baylor College of Medicine. And I have a lab at the Neurological Research Institute, which is a collaborative effort between the Baylor College of Medicine and the Texas Children's Hospital in Houston. So, my lab is working on a subset of lysosomal storage disorders that are, generally these diseases are inherited diseases that affect the metabolism of the cell and that often result in neurodegeneration. And in particular, we are studying Batten disease, also called neuronal ceroid lipofuscinosis. So, and in particular, one form of Batten disease, which is the juvenile form. There are many different types of neurodegenerative diseases, and especially the forms that are inherited from the parents. Typically, the parents are carriers, so they are not affected by the disease. But especially for the recessive diseases, 25% of their children would be affected. This is the case for the juvenile form of Batten disease. Both parents are carriers of mutations in the CLN3 gene, and one in four children inherit both mutations, and so they are affected by the disease. In Batten disease, what is missing is a protein that normally works in the lysosome. The lysosome is a substructure of the cell that works a little bit like a recycling center for the waste material of the cell. We can think of the cell like a city, that hosts a lot of different activities, and many of these activities just normally produce waste, and this waste must be acted on, must be collected, and must be either recycled or destroyed. And in the cell, the lysosome is the structure that performs these tasks. So actually the lysosome is the structure that is able to collect the waste material that is coming from the normal activities of the cell, and to degrade a little bit like a waste-based energy plant, or like recycling bins. So it can either degrade or recycle, or both, the components of this waste material. So in juvenile Batten disease, there are mutations in the gene that contains the instructions to build the CLN3 protein. The CLN3 protein is a protein that normally works in the lysosome. When this protein is missing, the lysosome no longer works correctly. And so over time, there is accumulation of this waste material. This accumulation never gets back. Over time, over years, it only becomes worse and worse and worse. And in particular, it will affect the normal function of the neurons, which are the cells that constitute our brain. And because the cell no longer can tackle this increasing accumulation of material, eventually it will give up. There are several pathways that are implicated in these diseases that are contributing to make the cell sick. But eventually what happens is that neurons will die, and this is what is leading to global neurodegeneration in juvenile Batten disease. And so patients will experience over the years, first of all, the loss of vision, then continuous loss of the ability to move in an autonomous way, and then to eat and drink, and then eventually there is continuous loss of this motor function, and also of more general brain functions. So eventually there is a premature death. Currently there is no therapeutic option for juvenile Batten disease other than palliative care. So we focused the research in my laboratory to ask the question, can we improve somehow the degradative processes in the cells of Batten disease patients? So we know that the lysosome is not working properly. Now we have this factor, TFAB, that acts as an enhancer of lysosomal processes. So, basically, if we have more TFAB or more active TFAB in a cell, then the lysosome will work better, and the degradative processes will be more efficient, and waste management will be more efficient. So we were looking for a way to make it happen, to find some drug that could activate TFAB that is present in the cell, and to make waste management more effective.